What is osmotic pressure? Osmotic pressure is the pressure required to prevent the flow of solvent through a semi-permeable membrane when there's a difference in solute concentration between two solutions. Let's visualize this concept. Here we have two solutions separated by a semi-permeable membrane. The left side has a high concentration of solute particles, while the right side has a lower concentration. The natural tendency is for water to flow from the low concentration side to the high concentration side to equalize the concentrations. Osmotic pressure is the external pressure that must be applied to prevent this solvent flow across the membrane. Importantly, osmotic pressure is a colligative property, meaning it depends on the number of particles in solution rather than their chemical identity. For example, a solution with 20 salt ions would have higher osmotic pressure than a solution with 10 sugar molecules, even though the sugar molecules are larger. To summarize, osmotic pressure is the pressure needed to prevent solvent flow through a semi-permeable membrane, and it depends on the number of particles in solution rather than their identity. The mechanism of osmosis occurs at the molecular level across a semi-permeable membrane. A semi-permeable membrane separates two solutions with different solute concentrations. The left side has a lower solute concentration, while the right side has a higher concentration of solute molecules. The semi-permeable membrane allows the smaller water molecules to pass through its pores, but blocks the larger solute molecules. This process continues until equilibrium is reached, or until osmotic pressure prevents further water movement. In summary, osmosis is the movement of water molecules through a semi-permeable membrane from an area of lower solute concentration to higher solute concentration, continuing until equilibrium is reached. Semi-permeable membranes are specialized barriers that allow certain molecules to pass through while blocking others. These membranes contain microscopic pores that act as selective gateways. The size of these pores determines which molecules can pass through the membrane. Small molecules like water can easily pass through the pores, while larger molecules are blocked. This selective permeability is crucial for many biological and industrial processes. Natural semi-permeable membranes include cell membranes found in all living organisms. These membranes are composed of a phospholipid bilayer with embedded transport proteins. Artificial semi-permeable membranes are engineered for specific applications. These include cellulose acetate membranes used in water filtration, dialysis tubing for medical applications, and polyamide membranes for reverse osmosis. Unlike natural membranes, artificial membranes often have a more uniform pore structure that can be precisely engineered for specific separation requirements. The Van't Hoff equation provides a way to calculate the theoretical osmotic pressure of a solution. The equation is expressed as pi equals I CRT. Pi represents the osmotic pressure, which is the pressure needed to prevent the flow of solvent across a membrane. I is the Van't Hoff factor, which accounts for the dissociation of solutes in solution. C is the molar concentration of the solute, measured in moles per liter. R is the universal gas constant, with a value of 0.08206 liter atmospheres per mole per Kelvin. T is the absolute temperature in Kelvin. This equation shows that osmotic pressure is directly proportional to concentration and temperature. Let's calculate the osmotic pressure of a 0.1 molar glucose solution at 25 degrees Celsius, or 298 Kelvin. Since glucose doesn't dissociate in water, the Van't Hoff factor is 1. Plugging all values into our equation, we get an osmotic pressure of 2.45 atmospheres. The Van't Hoff equation provides scientists and engineers with a powerful tool to predict and understand osmotic pressure in various solutions and biological systems. This fundamental equation connects osmotic pressure to basic solution properties, helping us understand osmotic phenomena across chemistry, biology, and medicine. In this example, we'll calculate the osmotic pressure of a zero point 0.1 molar sodium chloride solution at 25 degrees Celsius. The equation for osmotic pressure is pi equals ICRT. Let's understand what each variable represents. Sodium chloride dissociates completely in water, splitting into sodium and chloride ions. For our 0.1 molar sodium chloride solution at 25 degrees Celsius, let's identify the values for each variable. The Van't Hoff factor I equals 2 for sodium chloride because each molecule dissociates into two ions, sodium and chloride. Now let's substitute these values into our equation, pi equals ICRT. Substituting our values, we get pi equals 2 times 0.1 moles per liter times 0.0821 liter atmospheres per mole Kelvin times 298 Kelvin. Simplifying the calculation, the osmotic pressure equals 4.892 atmospheres. Let's convert this pressure to other common units. Converting to millimeters of mercury, we multiply by 760, giving us 3,718 millimeters of mercury. In kilopascals, we multiply by 101.325, resulting in 496 kilopascals. And in pounds per square inch, we multiply by 14.7, giving us 71.9 psi. Let's visualize what this osmotic pressure means. Here we have a semi-permeable membrane separating a sodium chloride solution from pure water. The calculated osmotic pressure of 4.892 atmospheres is the pressure needed to prevent water from flowing into the sodium chloride solution. Isosmotic solutions, also called isotonic solutions, have equal osmotic pressure across a semi-permeable membrane. When two solutions are isoosmotic, there is no net movement of solvent between them, creating a state of equilibrium. Let's visualize two isoosmotic solutions separated by a semi-permeable membrane. In isoosmotic solutions, the concentration of solutes is such that they create equal osmotic pressures. The mathematical representation of isoosmotic solutions is simply that the osmotic pressures are equal. In this state of equilibrium, individual particles continue to move randomly, but there is no net movement of water across the membrane. When a cell is placed in an isotonic solution, it maintains its normal shape. The concentration of solutes outside the cell is equal to the concentration inside, resulting in no net osmotic pressure. Water molecules move in and out of the cell at equal rates, creating a dynamic equilibrium. Isoosmotic solutions have important medical applications. A common example is 0.9% sodium chloride solution, also known as normal saline. Normal saline is isotonic with human blood, meaning it has the same osmotic pressure as blood plasma. This isotonic relationship is critical for intravenous fluids, as it prevents damage to blood cells that would occur with solutions of different osmotic pressures. To understand why 0.9% sodium chloride is isotonic with blood, we need to look at its concentration. Human blood plasma has an osmolarity of approximately 290 milliosmoles per liter, and normal saline is formulated to match this. Because normal saline is isoosmotic with blood, it can be safely administered intravenously without causing osmotic damage to cells. Hypoosmotic or hypotonic solutions have a lower solute concentration than a reference solution. This lower concentration results in lower osmotic pressure compared to the reference solution. When cells are placed in a hypotonic environment, the concentration gradient causes water to flow into the cells. Let's compare a reference solution with a hypotonic solution. Notice how the hypotonic solution has fewer solute particles. When a cell is placed in a hypotonic environment, water flows into the cell due to the concentration gradient. As water enters the cell, it begins to swell, increasing in volume while the solute concentration inside decreases. If too much water enters the cell, it may eventually burst. a process called lysis. This happens because the cell membrane cannot withstand the increasing pressure. Let's look at some real-world examples of hypotonic solutions. Distilled water is hypotonic to blood cells, which is why red blood cells burst when placed in pure water. Fresh water is hypotonic to marine organisms. This is why saltwater fish cannot survive in fresh water. Their cells would take in too much water and rupture. Sports drinks that are diluted too much with water can become hypotonic to your body's cells. In medical settings, intravenous fluids with salt concentrations less than 0.9% sodium chloride are considered hypotonic and must be used carefully. Hypotonic solutions have important clinical applications. They can be used therapeutically to hydrate cells when needed. However, hypotonic conditions can be dangerous, especially for brain cells, potentially leading to cerebral edema or brain swelling. They may also damage fragile tissues by causing excessive swelling, which is why proper osmotic balance is crucial in medical treatments. Hyperosmotic or hypertonic solutions contain a higher concentration of solutes than the reference solution. In a hyperosmotic environment, the solute concentration outside the cell is higher than inside the cell. This creates an osmotic pressure gradient that causes water to flow out of the cell through the semi-permeable membrane. As water leaves the cell shrinks and may undergo a process called crenation, where the cell membrane becomes irregular and wrinkled. The loss of water causes the cell to shrink, and in extreme cases, the cell membrane becomes wrinkled and irregular in a process called crenation. Examples of hyperosmotic solutions include concentrated salt solutions, seawater, 3% sodium chloride solution used in laboratories, and hypertonic intravenous solutions used in medical settings. Cells exist in fluid environments and must maintain osmotic balance to prevent excessive swelling or shrinking that could damage their structure. When a cell contains a higher concentration of solutes than its surroundings, water molecules move into the cell through osmosis. This inwardward water movement occurs because water flows from regions of lower solute concentration to regions of higher solute concentration. To prevent damage from excessive water movement, cells engage in osmoregulation, the active regulation of osmotic pressure to maintain proper cell volume and fluid balance. Cell membranes contain specialized transport proteins that help maintain osmotic balance. Different proteins serve different functions. Ion channels allow specific ions to pass through the membrane passively, following their concentration gradient. The sodium-potassium pump uses energy to actively transport sodium ions out of the cell and potassium ions into the cell against their concentration gradients. Aquaporins are specialized water channels that allow water molecules to move rapidly across the membrane in response to osmotic gradients. Different cell types have evolved specific adaptations to survive in their unique osmotic environments. Plant cells have rigid cell walls that allow them to maintain turgor pressure in hypotonic environments. Animal cells have flexible membranes and are more susceptible to osmotic changes. Bacterial cells often have cell walls and special adaptations to survive extreme conditions. Red blood cells provide an excellent example of how cells respond to different osmotic environments. In an isotonic solution, red blood cells maintain their normal biconcave shape. In a hypotonic solution, water flows into the cell, causing it to swell and potentially rupture in a process called hemolysis. In a hypertonic solution, water leaves the cell, causing it to shrink and develop a spiky appearance called crenation. Understanding osmotic pressure and how cells respond to osmotic environments is fundamental to cell biology and medical applications such as IV fluid therapy and dialysis. Plant cells rely on osmotic pressure to maintain their structure and rigidity. Unlike animal cells, plant cells have a rigid cell wall surrounding the cell membrane, with a large central vacuole filled with water and dissolved substances. Turgor pressure occurs when water moves into the plant cell through osmosis. Water molecules flow from areas of lower solute concentration to higher solute concentration across the semi-permeable cell membrane. As water enters, it creates pressure that pushes outward against the rigid cell wall. This pressure is called turgor pressure. Turgor pressure is essential for maintaining plant structure. It provides the rigidity plants need to stand upright. When plants lose water, turgor pressure decreases and the cells become less rigid. This causes the plant to wilt and droop. Let's compare a turgid cell with a plasmolyzed cell. A turgid cell is filled with water, pressing against the cell wall. In a hypertonic environment where the concentration of solutes outside the cell is higher, water leaves the cell through osmosis. This causes the cell membrane to pull away from the cell wall, a process called plasmolysis. Let's observe how a plant wilts and recovers. When a plant loses water, the cells lose turgor pressure and the plant begins to droop. When water is reintroduced, osmosis causes water to flow back into the cells, restoring turgor pressure. The plant becomes rigid and upright again. To summarize the key points about osmotic pressure in plant cells. Turgor pressure results from osmotic water movement into the cell. The rigid cell wall provides structure to withstand this pressure. When plants lose water, turgor pressure decreases, causing wilting. And in hypertonic environments, water leaves the cell, causing plasmolysis. Kidneys use osmotic pressure to filter blood and regulate water balance in the body. The functional unit of the kidney is the nephron, which consists of several specialized segments. The kidney creates and maintains a concentration gradient in the medulla, with increasing solute concentration toward the inner medulla. Blood filtration begins in the glomerulus, where water and small solutes pass into Bowman's capsule due to pressure gradients. In the proximal tubule, essential nutrients, ions, and water are reabsorbed into the bloodstream. The loop of Henle is crucial for creating the concentration gradient through a process called countercurrent multiplication. As fluid flows down the descending loop, water leaves via osmosis. In the ascending loop, sodium chloride is actively pumped out, but the tubule is impermeable to water. In the collecting duct, water reabsorption occurs via osmosis, following the concentration gradient established by the loop of Henle. The hormone ADH regulates water permeability in the collecting duct, allowing the body to produce either concentrated or dilute urine depending on hydration status. When ADH levels are low, the collecting duct remains relatively impermeable to water, resulting in dilute urine. When ADH levels are high, water is reabsorbed, producing concentrated urine. Through these osmotic processes, the kidneys precisely regulate water and solute balance, maintaining homeostasis in the body. Blood plasma is the liquid component of blood, making up about 55% of total blood volume. It consists of about 92% water, 7% proteins, and 1% other solutes, including electrolytes, nutrients, and waste products. Plasma proteins play a critical role in maintaining osmotic balance. Among these proteins, albumin is the most abundant and important. Oncotic pressure is a form of osmotic pressure, specifically created by plasma proteins. It's essential for maintaining proper fluid balance between blood vessels and surrounding tissues. Inside blood vessels, we have water, proteins, primarily albumin, and various ions and small molecules. These proteins cannot easily cross the vessel wall. Two main forces regulate fluid exchange: hydrostatic pressure, blood pressure, pushing fluid out, and oncotic pressure, pulling fluid back in. Blood plasma maintains a delicate osmotic balance. When this balance is disrupted, it can lead to medical conditions. Edema occurs when oncotic pressure is too low, often due to reduced albumin levels in conditions like liver disease or malnutrition. This causes fluid to leak into tissues, resulting in swelling. Conversely, dehydration can increase the concentration of proteins in plasma, raising oncotic pressure and pulling more fluid from tissues into the bloodstream, further dehydrating the tissues. Let's summarize the key points about blood plasma and osmotic balance. Blood plasma 92% water, 7 proteins, 1 other solutes. Albumin contributes 60-80% of oncotic pressure. Normal oncotic pressure is 25 to 30 mm/Hg. Balance between oncotic and hydrostatic pressures regulates fluid movement between vessels and tissues. Low albumin levels lead to edema in tissues. Dehydration increases protein concentration and oncotic pressure. Neurons rely on specific ion concentrations and osmotic balance to function properly. The neuronal membrane separates different ion concentrations. Inside the cell, potassium is high, while sodium and chloride are low. These ion channels regulate the movement of specific ions across the membrane. These ion concentration differences create the resting membrane potential of about -70 millivolts. When a neuron is stimulated, it generates an action potential, a rapid change in membrane voltage that propagates along the axon. Osmotic imbalances can severely disrupt neuron function. In normal conditions, ion concentrations are carefully regulated. In a hypoosmotic environment, water flows into the cell, causing it to swell and diluting ion concentrations. In a hyperosmotic environment, water leaves the cell, causing it to shrink and concentrating ions. These osmotic imbalances lead to various neurological symptoms. Hypoosmotic conditions can cause seizures, while hyperosmotic conditions can lead to confusion and impaired neural function. In summary, proper osmotic balance is critical for maintaining normal neuronal function and preventing neurological disorders. Osmotic pressure plays a crucial role in the digestive system, driving water movement and nutrient absorption across the intestinal walls. Let's examine how osmotic gradients function throughout the digestive tract. The small intestine is where most osmotic absorption occurs, with specialized structures called villi increasing the surface area. Let's look at a cross-section of the intestinal wall to see how villi increase surface area for absorption. Let's zoom in on a single villus to understand how osmotic pressure drives absorption. The intestinal epithelium acts as a semi-permeable membrane. Nutrients, ions, and water move across this barrier through various mechanisms. Nutrients and sodium ions create an osmotic gradient across the membrane. As these particles are actively transported from the intestinal lumen into the bloodstream, water follows by osmosis to balance the concentration difference, moving from an area of lower solute concentration to higher solute concentration. When this balance is disrupted, conditions like osmotic diarrhea can occur. Let's compare normal intestinal absorption with osmotic diarrhea. In normal digestion, nutrients are absorbed across the intestinal wall, and water follows due to the osmotic gradient. In osmotic diarrhea, poorly absorbed solutes like lactose in lactose intolerance or artificial sweeteners remain in the intestinal lumen. These solutes draw water into the intestine through osmosis, resulting in watery stools and diarrhea. Several clinical conditions involve osmotic imbalances in the digestive tract. In lactose intolerance, undigested lactose remains in the intestine, drawing water in by osmosis and causing diarrhea. Magnesium-based laxatives intentionally create an osmotic gradient to increase water content in the stool. Sugar alcohols found in sugar-free foods can also cause osmotic diarrhea when consumed in large amounts. Intravenous fluid therapy is a critical medical intervention that relies on understanding osmotic pressure to safely deliver fluids directly into the bloodstream. When administering IV fluids, healthcare providers must carefully consider the osmotic pressure of the solution relative to blood plasma. The osmotic pressure of blood plasma is approximately 290 milliosmoles per liter. This pressure affects how fluids move between blood vessels and surrounding tissues. IV solutions are classified into three main types based on their osmotic pressure relative to blood plasma. Isotonic solutions have the same osmolarity as blood plasma, approximately 290 milliosmoles per liter. They cause no net fluid shift between blood vessels and cells. Hypotonic solutions have lower osmolarity than blood plasma. When administered, water moves from the bloodstream into cells, causing them to swell. Hypertonic solutions have higher osmolarity than blood plasma. They draw water out of cells into the bloodstream, causing cells to shrink. Isotonic solutions like 0.9% sodium chloride are commonly used for volume expansion, treating dehydration, and as a medium for blood transfusions. Hypotonic solutions such as 0.45% sodium chloride are used to treat cellular dehydration, hypernatremia, and hyperosmolar states. Hypertonic solutions like 3% sodium chloride are beneficial in treating cerebral edema, hyponatremia, and increased intracranial pressure. Administering IV fluids with incorrect osmolarity can lead to serious medical complications. For example, using an isotonic solution when a hypotonic solution is needed may fail to correct cellular dehydration, allow hypernatremia to persist, and potentially cause fluid overload. Similarly, administering a hypotonic solution when a hypertonic solution is required can worsen cerebral edema, exacerbate hyponatremia, and even cause hemolysis of red blood cells. Let's examine some common IV solutions used in clinical practice and their osmotic properties relative to blood. Normal saline, or 0.9% sodium chloride, is an isotonic solution with an osmolarity of 308 milliosmoles per liter. It's commonly used for volume replacement and treating dehydration. Lactated Ringer's solution is also isotonic with an osmolarity of 273 milliosmoles per liter. It's frequently used for fluid resuscitation and in treating burn injuries. D5W, or 5% dextrose in water, is hypotonic with an osmolarity of 252 milliosmoles per liter. It's used to provide free water and treat hypernatremia. Hypertonic saline, or 3% sodium chloride, has a high osmolarity of 1,026 milliosmoles per liter. It's used in treating severe conditions like cerebral edema and severe hyponatremia. Regardless of which IV solution is used, careful monitoring of electrolytes and fluid balance is essential to prevent complications and ensure optimal patient outcomes. Dialysis is a life-saving medical procedure used when a patient's kidneys fail to adequately filter waste from the blood. Normally, healthy kidneys filter blood by removing waste products and maintaining fluid and electrolyte balance. In kidney failure, waste products accumulate in the blood, which can be life-threatening without intervention. Dialysis uses a machine that acts as an artificial kidney. Blood is removed from the patient, filtered in the machine, and then returned to the patient. The key component of dialysis is the semi-permeable membrane, which allows some substances to pass through while blocking others. Blood contains various components, including waste molecules, essential blood components like cells and proteins, and electrolytes and water. The principle of osmotic pressure drives the dialysis process. The membrane is designed to allow smaller waste molecules and excess water to pass through while retaining larger essential blood components. During dialysis, waste products move from the blood through the membrane into the dialysate solution due to the concentration gradient. The dialysate solution is specifically formulated to create the right osmotic environment for waste removal. Dialysis has several important clinical applications in treating kidney failure. The most common use is treating end-stage renal disease, but dialysis is also used for acute kidney injury, toxin removal, correcting fluid and electrolyte imbalances, and as a bridge to kidney transplantation. To summarize, dialysis relies on several key principles: a semi-permeable membrane for selective filtration, osmotic pressure as the driving force, concentration gradients to control solute movement, and carefully formulated dialysate solutions. Reverse osmosis is an industrial water purification technology that applies pressure to overcome natural osmotic pressure. To understand reverse osmosis, let's first look at natural osmosis, where water naturally moves from low to high solute concentration. In normal osmosis, water molecules move across the membrane from an area of low solute concentration to high solute concentration. In reverse osmosis, external pressure is applied to overcome the natural osmotic pressure, forcing water in the opposite direction. A typical reverse osmosis system consists of several key components. Feed water enters a high-pressure pump, which forces it through a semi-permeable membrane. The membrane allows water molecules to pass through while blocking contaminants like salts, bacteria, and other impurities. The clean water or permeate is collected, while the concentrate containing rejected contaminants is discharged. The pressure forces water molecules through the membrane, separating them from contaminants. This produces clean water while concentrating the impurities in the waste stream. Reverse osmosis has several major industrial applications. The most notable is desalination, which converts seawater into fresh water by removing salt and other minerals. In municipal water treatment, reverse osmosis removes contaminants, bacteria, and viruses from drinking water supplies, improving water quality, taste, and odor. Industries use reverse osmosis to recycle processed water, treat wastewater, and concentrate valuable compounds, reducing both resource consumption and disposal costs. To summarize, Reverse osmosis is a pressure-driven membrane process that overcomes natural osmotic pressure to purify water. It's a versatile technology with applications spanning from desalination to industrial water recycling. Food preservation through osmotic pressure is one of the oldest and most effective methods of extending food shelf life. When microorganisms encounter a hyperosmotic environment rich in salt or sugar, water is drawn out of their cells through osmosis. This dehydration causes the microbial cells to shrink, deactivates their enzymes, inhibits their growth, and ultimately leads to their death. Traditional food preservation methods leverage osmotic pressure to extend shelf life. Many popular preserved foods rely on osmotic principles. Beef jerky uses salt to draw out moisture. Fruit jams use sugar's osmotic properties. Pickles combine salt and acid in a brining process. While traditional osmotic preservation methods have been used for millennia, modern food technology has refined these techniques with precise control of water activity and combined preservation approaches. In summary, osmotic food preservation creates environments where water is drawn out of microorganisms, preventing their growth and extending food shelf life using natural principles. Osmotic pressure is a fundamental force shaping life in natural environments. It determines where organisms can live, drives evolutionary adaptations, and influences biodiversity patterns across different ecosystems. Aquatic organisms face distinct osmoregulatory challenges depending on their environment. In freshwater environments, fish cells face a hypotonic situation where water tends to flow into the cell. Freshwater fish must constantly pump out excess water and actively retain ions to maintain osmotic balance. Conversely, in saltwater environments, fish cells are in a hypertonic situation where water tends to flow out of the cell. Marine fish must drink seawater and excrete excess ions through specialized cells in their gills. Many aquatic organisms have evolved specialized adaptations for osmoregulation in their specific environments. Salmon are remarkable for their ability to transition between freshwater and saltwater environments during migration. They have specialized chloride cells in their gills and hormone-regulated osmoregulation that can switch modes. Some organisms have evolved remarkable adaptations to survive in environments with extreme osmotic conditions. Halophilic microbes thrive in extremely salty environments, like the Great Salt Lake, with internal salt concentrations that would kill most organisms. Desert frogs, like the Australian water-holding frog, can store water under their skin and in their bladder, allowing them to survive extended dry periods. Osmotic pressure has significant ecological implications, particularly as environments change. Changes in salinity due to climate change or human activities can stress organisms beyond their adaptive capacity, potentially reducing biodiversity. There are several laboratory methods for measuring osmotic pressure, each with specific applications and limitations. Osmotic pressure can be measured using both direct and indirect methods. Direct measurement uses osmometers, which typically consist of a chamber divided by a semi-permeable membrane. The pressure required to prevent water flow across the membrane equals the osmotic pressure. Membrane osmometers directly measure the hydrostatic pressure needed to halt osmosis. Colloid osmometers are specifically designed to measure oncotic pressure from proteins in biological fluids. Indirect methods utilize colligative properties like freezing point depression and vapor pressure lowering to calculate osmotic pressure. Freezing point osmometers are most commonly used in clinical laboratories. They measure how much the freezing point of a solution is lowered compared to pure water, then convert this to osmolality, expressed in milliosmoles per kilogram. Measuring osmolality is crucial in clinical settings for assessing fluid and electrolyte balance. The normal serum osmolality ranges from 275 to 295 milliosmoles per kilogram. Clinical applications include assessing hydration status, diagnosing electrolyte disorders, monitoring conditions like diabetes insipidus, evaluating kidney function, and detecting toxic ingestions. Modern laboratory equipment for measuring osmotic pressure includes freezing point and vapor pressure osmometers. These devices offer automated sampling and measurement, providing results in just 1 to 2 minutes. They typically require only a small sample volume, usually between 20 and 50 microliters. To summarize, osmotic pressure can be measured through both direct methods using membrane osmometers and indirect methods using freezing point depression or vapor pressure lowering. These measurements provide critical information for clinical assessment of fluid and electrolyte balance. Osmotic pressure principles have found applications across numerous scientific and technological domains. In biology, osmotic pressure regulates cell volume, drives water transport in plants, and facilitates nutrient absorption across membranes. In medicine, osmotic pressure principles are applied in intravenous fluid therapy, dialysis treatments, and advanced drug delivery systems. Industrial applications leverage osmotic pressure for water desalination through reverse osmosis, food preservation, and even clean energy generation. Environmental applications include water quality monitoring, soil moisture management, and maintaining ecosystem balance. Several emerging technologies are leveraging osmotic principles in innovative ways. Osmotic power generation, or blue energy, harnesses the osmotic pressure difference between fresh and salt water to generate clean electricity. Smart drug delivery systems use osmotic principles to control the release rate of medications, improving treatment efficacy and reducing side effects. Future directions in osmotic pressure applications include developing nanoosmotic technologies, biomimetic membranes that mimic natural systems, AI-optimized osmotic processes, and self-regulating implantable medical devices. Understanding osmotic pressure continues to drive innovations that address global challenges in healthcare, energy production, and environmental sustainability. Let's summarize the key concepts of osmotic pressure. Osmotic pressure is the pressure required to prevent the flow of water across a semi-permeable membrane from a region of lower solute concentration to a region of higher solute concentration. The Van't Hoff equation, pi equals I times C times R times T, mathematically describes osmotic pressure. Here, pi represents osmotic pressure, I is the Van't Hoff factor accounting for dissociation, C is molar concentration, R is the gas constant, and T is absolute temperature. Solutions can be classified based on their osmotic pressure relative to a reference solution. Isoosmotic solutions have equal concentrations of solutes, resulting in no net water movement. Hypoosmotic solutions have lower concentrations of solutes, causing water to flow into cells. Hyperosmotic solutions have higher concentrations of solutes, drawing water out of cells. Osmotic pressure plays critical roles in various biological systems. It regulates cell volume and homeostasis, maintains plant cell turgor, drives kidney filtration, balances blood plasma, and supports nerve cell function. The principles of osmotic pressure are applied in numerous practical technologies. These include medical applications like IV fluids and dialysis, industrial processes like reverse osmosis, food preservation, and environmental technologies like desalination. All these concepts interconnect in a framework centered around osmotic pressure. At the center is osmotic pressure itself, connected to the mathematical Van't Hoff equation, different solution types, biological significance, practical applications, measurement methods, and factors affecting osmotic pressure. In conclusion, osmotic pressure is a fundamental physical principle that underlies countless natural processes and technologies. Understanding this concept is essential in fields ranging from cell biology to industrial applications.